Thank you for the introduction. So in this webinar on effective preclinical solutions to tackle oncology drug resistance, I'll start by briefly discussing the evolving landscape of cancer drug resistance and the clinical need and how it can be challenging to model preclinically. And then how the four-step strategy can be used to navigate solutions appropriately. And I'll give some examples of different models to demonstrate what can be achieved, the key features, challenges, and opportunities. So if we look at cancer drug resistance as a whole, it's actually multifaceted, as described in this paper in Nature. And initial success in cancer remission that is widely achieved with many different therapeutics, it becomes challenging because of disease relapse as a result of resistance. And you can break down the complexity into basic determinants of resistance around the therapeutic and its target, the tumor, and also the environment. So, for example, the size of the tumor and the growth rate and the heterogeneity with the different genomic alterations that reside within the tumor are primary sort of determinants of resistance. And then there's also the physical barriers, the immune system, and the tumor microenvironment, which provides alternative non-target related means of resistance. And then there are target-specific or there's impact of the therapeutic pressures that are influencing the landscape of resistance. So, for example, to overcome chemotherapy or radiotherapy resistance, more targeted approaches have been developed, which have resulted in new challenges with the emergence of the resistance. And then there's also new types of targets within the immune system, and the new therapeutics like immune checkpoint inhibitors, cellular therapies, PROTACs, which means that the cancer will and can evolve and adapt further. So it makes the whole sort of picture more complicated and evolves as well with time. So in this paper in Nature Reviews Drug Discovery, this really highlights the types of new drugs that have reached the clinic in the last 2 decades. Cytotoxic drugs are still playing a role but the largest contribution is coming from targeted agents, especially kinase inhibitors, and then the targeted biologics, which are dominated by immune checkpoint inhibitors. And then on the right, there's the different types of agents shown down here, which have been approved in the sort of last 20 years. And you can see that the more recent agents, which are shown in red, and then the blue ones, which are in the sort of early 2000s. And again, the kinase inhibitors have been dominating this throughout the past 2 decades. But you can see how immune checkpoint inhibitors, targeted as cytotoxic agents like ADCs, PARP inhibitors, autologous cell therapies, bispecifics, all these approvals have been more recent in the sort of last 5 to 10 years. And then the emergence to resistance is already evident for many of these, and therefore there's a need or a requirement for new strategies to combat these new cancers or these new resistance. And as such, the preclinical models must evolve too to mimic this new landscape. As resistance can be multifaceted, one model may not be enough to interrogate it, but having a preclinical toolbox that can help to differentiate therapeutic approaches is shown here in our sort of Crown Biosciences capabilities. So when it comes to evaluating some of these strategies in these models, it can actually be very difficult to know where to start and which models will be useful in answering specific questions, whether that's in vitro or in vivo. And there's also a lot more options and new technologies that have emerged to improve the predictive value of these models and the patient relevance compared to some of these initial earlier models that were were developed to address cancer many years ago. These are now being overtaken by new in vitro and in vivo and ex vivo models. So, for example, there's the emergence of biobanks of patient-derived models over the last few decades. Some new, so the new models like the PDX, and then you've also got the organoids, which are able to better predict clinical response and biomarker identification. There's also advances in culturing, assay conditions, and data acquisition for complex in vitro biology. And this can provide valuable insights in the sort of early drug discovery sort of stages, as well as being able to screen patient tissue directly and early on, but also directly by using ex vivo tissue from the patient directly. With the emergence of new immunotherapeutics, the demand for immune oncology relevant models became very high, and engineering technology like CRISPR modifications has also enabled new models to be developed. Again, this has really impacted and improved some of the immune deficient nature of mice to allow better engraftment to create new models, such as the humanized systems. There's also knock-in, knock-out strategies, which can be developed, and targets can be humanized. There's also animal imaging that enables more complex biology to be mimicked, especially with those later stages of diseases. So when you're looking at orthotopic or metastatic modeling, and this really does enable a lot more sort of opportunities in understanding the disease biology and progression of cancer. So some of these models I'll describe or give examples of in the context of resistance, but you also can find a lot of the model information in our databases, which are just listed here. To assess resistance, as I mentioned, there isn't one perfect preclinical model that can mirror all the different aspects of resistance. And so therefore you do have to approach this from an integrated preclinical approach, so that the different sort of features and the phenotypes can be measured appropriately, and you look at efficacy and toxicity in the most patient relevant system as early on as possible. And this may involve different types of systems in vitro and in vivo, as well as looking at identifying potential biomarkers of response or resistance. So hopefully, like I said, some of the examples will dive into this a little bit more. So this brings me to the 4-step strategy to investigate drug resistance using these preclinical tools. So the first step is what is resistance? What's the resistant phenotype of interest? And now this may seem an obvious question, but being actually specific about the clinical response or resistance profile needed to target therapeutically will actually help determine whether this can be modeled preclinically or what questions need to be answered sufficiently to draw the required outcomes and conclusions. The second step is then to find this a model. So are there any acquired or intrinsic resistant models already available that could be used for this purpose? And in general, acquired resistance is what is preferred, but in some cases, intrinsic models may be sufficient. So search and select the most appropriate preclinical model to really recapitulate that clinical phenotype of resistance as outlined in step 1. Now, in some cases, the resistance model may not be available. So there may be an opportunity to improve it. And so can that resistance model, the ideal resistant model, be created? And this is, again, sort of a complex sort of approach, especially because sometimes the data is needed urgently, but it may be the only option, especially if there's no models readily available, and especially with new emerging targets and new therapeutics, it's important to make sure that the model is appropriate. And then finally, characterizing the resistance, not just pharmacologically, but also applying sort of deep learning to interrogate any responder, non-responder profiles, which can then potentially identify new strategies to overcome resistance. There may be combination approaches that might be more appropriate, but also potential biomarkers and patient stratification, which really can produce the best success for that mechanism. So let's start with step 1. So some of this was nicely introduced in that Nature paper I showed, and also how multifactorial it is via sort of different mechanisms. And we touched on the various standard care agents or new agents and the first or second line treatments and therapeutics that are available for different cancer targets at different stages of the disease. So initially there was a very good response, and I think That is fantastic with the range of different drug development that's going on at the minute. But eventually resistance emerges, which tips the balance, causing tumor relapse and disease progression. Some of these factors can cause a drug to fail. It can be due to that the cancer is adapting or acquiring mechanisms to the therapeutic pressures applied in the sort of short term and long term. It can be due to drug-related factors such as uptake, efflux, or inactivation. There may be alterations to the target or pathway or dominant clone, and also mechanisms preventing cell death through external means or internal mechanisms. So these will require different types of models. And again, not one model won't be able to mimic all of this. And then in terms of intrinsic versus acquired resistance, either existing before treatment, so that's the intrinsic resistance, or generated after therapy, which is the acquired. It's responsible for most relapses of cancer and obviously leads to the death of patients. For intrinsic resistance, there may be some responses initially, or none at all, which is shown on the left-hand side. Whereas for acquired, there is an initial response, which is shown on the right. And eventually this is lost and there is tumor relapse. And then alternative strategies may need to be implemented, so a change in treatment to tackle the resistance, which is the next step. And this may be guided by biomarkers, for example, but also the initial predictive biomarker with the first treatment may provide the alternative or best strategies to actually prevent any resistance emerging, which could be also valuable. So those predictive biomarkers play an important role in making sure that the resistance is overcome. The second step, which is to search for the right model. So you can do this by searching databases for models that are already available. So this could be intrinsic models in cell lines or PDX, and then also acquired models of resistance from the clinic. And you can use actually our database. So this is our complementary model database, which can help find some of those models. There's 4 main databases, one for PDX, one for organoids, one for murine models, and then the fourth one for xenograft cell line models. And this is a centralized model repository, which is extensive sort of model characterization and collection. So, for example, there's over 2,400 PDX, there's over 600 organoids. Some of these organoids have matched xenograft models. There's a diverse collection of murine models, and over 800 cell lines with over 240 different xenograft models. So a huge selection. And these are all categorized to cancer type and can be filtered when you are doing your search, just to make it easier to explore the models. And this is a great starting point to find the right model. And the characterization, which you can see is in the video running there, that there is, it's quite an intuitive database. So you can scroll through the different sort of features and the different sort of data that's included. So the characterization of the models will be genomics, proteomics, there's be growth, and then more some model-specific information, such as patient information. So this actually is, this slide is just a high-level categorization of some of those resistant models at Crown. And this is across cell lines, PDXs, in vitro and in vivo, as well as tissue. And like I said, I'll try and give you some examples of each one, but there's actually quite a lot really to share today. So again, there's more information in the databases, or you can reach out to us to ask specific questions. So some of these are straightforward cell line chemo-resistant models. So we've got things like A278T cisplatin resistant ovarian models. There's also numerous PDX models with resistance to chemotherapeutics. There's also different targeted agents as well. So, for example, you've got Gleevec resistant models, niraparib resistant, osimertinib, and also multi-drug resistant lines as well. We also have a large number of models being developed or characterized, and you can again, if you don't see a model, you can again ask, and we can let you know what the status of some of those model development is. So we do also have some very unique models or large collections. So, for example, we have a biobank of clinical specimens. These have clinical history, which, there's actually currently almost 1 million different patient samples. So this is an extensive sort of collaboration of clinical network across 60 sort of different entities in the US, Europe, and Asia, where we've been able to accumulate all the different sort of tissue types. We also have a large collection of organoids, which have been biobanked, and these can also be used for resistance modeling as well. And actually, we've just released a screening platform called OrganoExplore, which uses our sort of innovative organoid cryopreservation technology and also assay automation and screening, so that we can look at a large panel of organoids, as well as specialized panels such as KRAS mutation, which were recently presented on and showed the response to AMG 510 at the EORTC AACL meeting last month in Boston. If you click on some of the models here, so if we look at the PDX models from pre-treated PDX, you can see that so these are referred to as the next generation of PDX models, as these are from patients who have undergone treatment to first, second, or third line treatment in the last decade. And these are, so they're reflecting some of the new sort of approaches, new treatments and new therapeutics, some of which have just come to market, as mentioned earlier. And so these patients have very aggressive late-stage cancers that have relapsed from multiple therapeutic strategies. The diagram on the left shows the spread across different cancer types. And bearing in mind this is an ongoing collection, so the numbers of models will continue to grow. And then on the right is a summary of some of the treatments. So still a lot of chemo in there, as that is typically the first-line treatment for many cancers. But there's also combinations, targeted agents. The unspecified that's mentioned here are investigational drugs. So the information on these is not released yet. And these patients are enrolled as they've failed all other types of treatments. And as you can see, there are a number of models that are also coming from patients who received immune checkpoint inhibitors and other immunotherapeutics. And also hormone therapies, such as those for breast cancer, prostate cancer. And actually, prostate cancer is still difficult to generate as a PDX, so it's nice to see more in this collection, and that's because these tumors are typically very aggressive. So we have a number in the pipeline that are going through expansion so that we can assess the treatment and the resistance phenotype. So here's an actual summary of those prostate PDX models from patients who have received standard care treatment. So a typical sort of clinical landscape is just shown here with the different sort of treatment options based on the Gleason score and the staging, and then how the tumor can relapse and the next treatment options based on hormone sensitivity. But most hormone-dependent cancers become refractory after 1 to 3 years, and the growth resumes despite this hormone treatment that's given. And they were previously considered hormone-refractory prostate cancer or androgen-independent prostate cancer, but actually the term castration-resistant prostate cancer is more accurate and more commonly used now. As it's not responsive to castration treatment, whether that's chemically induced or surgically induced. And in this stage, really, docetaxel has shown the best survival benefit for patients, but also hormonal therapies are still used, as many castrate-resistant prostate cancers still show reliance on androgen receptor activation. So agents like abiraterone and enzalutamide, which is a second-generation antagonist of androgen receptor, has been useful in the treatment of castrate-resistant prostate cancer. But again, the tumors can relapse and also metastasize to various organs, and particularly to lymph nodes and to the bone. And this poor survival is associated with the sort of metastatic castrate-resistant prostate cancer, with the survival time of only approximately 16 to 18 months, with the bone mass causing death in most of those patients. So metastatic castrate-resistant prostate cancer is really an area of high unmet need in the clinic. And here, having new PDX models which have been created from these patients are highly valuable. So the top three are derived from the primary site. So this is following radical prostatectomy, whereas the others are derived from the metastatic site. So you've got different sort of representation of the stage of that disease. And all of these patients have had prior treatments, such as hormonal treatment, chemotherapeutic, targeted agents, such as enzalutamide. And actually, one of the models, which is shown here on the top right, PR6511, which is from a metastatic castrate-resistant prostate cancer patient, was actually confirmed to have a TMPRSS ERG fusion, P10 loss, and also high levels of PSA expression. And as you can see from the graph, there was no significant response observed following treatment with enzalutamide or abiraterone in mice bearing this sort of subcutaneous PDX model of this patient. So this table shows various other models of colorectal cancer with KRAS mutation, which again is a huge area of interest. And this is, I mean, because it's one of the most frequently mutated oncogenes and has been recognized as an undruggable target for many years. So here, the castrate-resistant, the colorectal patients are receiving first-line treatment of FOLFOX. There's also some combinations of Avastin in second line or third line. Specific targeted agents for G12C inhibition inhibitors are already being given to some of these patients, and these were recently approved, for example, for non-small cell lung cancer. But here in colorectal cancer with those KRAS mutations, these are listed as investigational drugs. So again, emerging sort of data can be drawn from these types of PDX models. So in this particular model that's highlighted, so there's a series of models that have been collected over different time points. So this is from a Caucasian female patient with metastatic adenocarcinoma. And this harbors a G12C mutation. And there was a good response initially, but this soon relapsed. And so biopsies were taken from different sort of stages, from liver metastasis. And then the patient was eventually progressed onto a fourth investigational drug. So this is a KRAS inhibitor resistant model that we're actually characterizing at the minute. So here you can see the growth profile of the subcutaneous G12C colorectal PDX models from the two relapse models that come from the same patient, but they're obviously at the different stages of the disease progression and treatment relapse. Both show weak response to AMG 510 and partial response to MRTX, but they both represent progressive disease and as such are classified as resistance to both agents. And now the original model was responsive to KRAS inhibitors and showed good TGI in the PDX, so that's not shown here, but there wasn't complete tumor regression. So again, that is due to the heterogeneity of that tumor. So these serial models of specific oncogenic mutations, response, and then relapse are really valuable for understanding that disease resistance and progression. And these are undergoing further characterization to understand the mechanism of that resistance. Other examples of resistance phenotype of high interest are shown in this table, and these have been generated from patients who have been treated with checkpoint inhibitors and then obviously become resistant. So again, these are currently undergoing profiling. There's quite neat models. There's some very interesting ones here, especially because they've been treated with pembrolizumab and nivolumab, and they're being characterized for in vivo growth as well as genetic profiling. So as these models can take some time to develop, as they're relatively new from the clinic, so it can take time to not only acquire them, but also to characterize and grow up the models. So models where the resistance has been engineered into some of our cell line models, for example, so, have proved very useful in the meantime. So, for example, we have a number of models with KRAS G12C mutation engineered into MIA-PACA2, which is a pancreatic cancer xenograft, which is just in under this tab. But for other targets, we have induced resistance in our PDX through continuous dosing in vivo. So, for example, I'm just going to give you an example of the RET inhibitor induced PDX model here. Just to kind of show you that the rationale for the design of this particular one. So there is a need for this type of resistance. Activating RET alteration, which is induced by gene fusions or point mutations, actually occurs in many different solid tumor types, including non-small cell lung cancer, breast cancer, and colorectal cancer. And actually, just very recently, 2 RET Pacific tyrosine kinase inhibitors were actually approved by the FDA for the treatment of thyroid cancer and non-small cell cancer. And there's also some publications shown here of some of the models from our colorectal cancer collection that were used in the studies. So despite the sort of encouraging efficacy to these RET inhibitors, acquired resistance such as solvent front RET G810R, S, or C mutations occur, and this has really limited their clinical benefit. So these are sort of on-target drug resistance. There's also off-target resistant mechanisms that have also been reported. So therefore, to kind of better understand and better investigate novel therapeutic strategies which can overcome these sort of acquired resistance for these novel agents, we look to establish PDX models from our colorectal collection by inducing the drug resistance by exposure to these specific agents. So subcutaneous PDX models were treated with 3 mg/kg dose of LOXA292. And then any tumors that outgrew under treatment were isolated, replanted, and then dosed again with 3 mg/kg or 10 mg/kg. And 2 of those colorectal models are shown here on the right. So each one have acquired different types of mutations. So you've got the V804M and the G810S. And also with these, you can see that the resistance profile is different as well. So there is, a more resistance shown with the gatekeeper mutation. The gatekeeper mutation still seems sensitive in one of these models. So actually, in that second publication just shown here, the group did also use other types of models for resistance profiling. So this is the BAFS lines of models that have been engineered to have specific mutations, so the BAF3 cell lines. So if you look here, we actually do have a collection of these lines in-house that we've generated as well. And again, these cover different types of targets, and these are very sort of custom built, mutated novel cell lines generated with those mutations in mind, which are then characterized in vivo. So this mouse propy cell line is actually really useful for developing these types of resistant models. It's dependent on the interleukin-3 for survival and proliferation. Upon transduction of those driver genes, BAFF3 cells become independent of interleukin-3, but dependent on that sort of transduced driver gene. So this has been a very popular system to generate models with those oncogene dependencies and vulnerabilities to specific targeted therapies. And these can be used to sort of estimate the oncogenicity of those driver mutations or the efficacies of different targeted drugs. And so they have been used quite broadly and quite widely, and this is nice to have sort of in our collection as well to help with some targeted agent investigations. So again, we can also look at other types of models of resistance. So if we look at the sort of metastasis models, just to mimic the late stage of a disease, which can actually emerge despite numerous different agents being applied to those cancers. And these can be quite difficult to model, just because the sort of mouse model requires surgery, and to model sort of distant metastasis, there's obviously different tumor microenvironments. So we do actually have to design the sort of model around that tumor microenvironment. So this, in this example, this is breast cancer, and this shows how different stages of the disease can be mimicked in mice. The tumors are implanted, for example, in the mammary fat pad of a mouse, and this can be used to model invasion and spontaneous metastasis to distant sites. And then you've got bone, experimental bone metastasis models or brain metastasis models that can also be implemented. So in this triple negative breast cancer model, the MDNB231, this is a bioluminescent engineered line. It makes it easier to actually track these models when they're put orthotopically or metastatically. And in this one, in the mammary fat pad, it's injected into the fourth mammary pad, and optical imaging is used to quantify the growth of that tumor. The model duration is quite short, it's about 3 to 4 weeks. And it's actually driven by that primary mammary fat pad lesion, because as the size reaches a certain limit, you have to euthanize. But it can be, it is validated with standard of care agents for triple negative breast cancer, which is docetaxel. And the incidence of metastasis is quantified at end stage. And the frequency of METs also in different cohorts of mice can be quantified as well. And you can see from these pie charts that the METs to the lung or the lymph nodes is quite dominant, whereas metastasis to other organs such as the brain or bone less so. And that is just normally because there's not sufficient time to develop those metastases at those distant organs. So as the instance of bone met in that is very low, the option would then be to maybe have a experimental or seeding model of metastasis. And this is the intracardiac model, where the MDNB231 cells are implanted into the left ventricle of the heart. It enters the circulation and lodges into the end plates of long bones, the jaw, the ribs, and the spine. And so within 3 weeks, you actually form lytic lesions similar to what you would see in the clinic. The metastases are monitored in real time, so you can see them in the long bones within 5 days. So it's quite a short model. The lytic lesions can be quite aggressive, and you can quantify those as well by X-ray or by MRI. And then the standard of care for bone metastasis, which is permethrinate, can also be used in these models quite nicely to show significant reduction in those lytic lesions. The untreated mice is shown here on the left, and these are, just play the video for you. So these are sacrificed 3 to 4 weeks, and the endpoint is driven by those lesions in the long bone, which is shown by the arrows. And this coincides with the sort of loss in clinical condition as well. So this is quite useful in terms of the bioluminescent imaging can be co-registered with the micro CT, so you can really kind of see exactly where those bone lesions are occurring. And then in the Taxotere treated group on the right-hand side, you can see how it limits the progression of the long bone lesions, reduces the loss in clinical condition, and actually extends the survival to 8 weeks. But however, due to the sort of inability of Taxotere to cross the blood-brain barrier, you do actually get lesions in the central nervous system and the brain developing. So you can see some in the spine as well as the brain, which have escaped that treatment. And so this now drives endpoint. So this again is reflective of the clinic. You will see the occurrence of brain lesions as a complication of late-stage metastatic breast cancer, which escapes docetaxel treatment. So this is actually quite a valuable model then for identifying suitable combination approaches, obviously with Taxotere, to treat metastatic breast cancer, which has progressed to the brain. And this actually wouldn't have been possible to measure without the imaging to kind of understand where some of those more resistant resistant tumors were occurring. You can also model directly in the brain microenvironment, which again is a metastatic site for many different types of cancers, so breast and lung amongst others. And the sort of presence of that blood-brain barrier does pose, a significant challenge for many different therapeutic types. So again, having these orthotopic implanted models as another example of experimental metastasis models is quite useful. So here, this example is the EML4-ALK fusion, which actually occurs in a small percentage of patients with non-small cell lung cancer, which actually typically responds really well to specific ALK inhibitors like crizotinib, in comparison to some of the other standard first-line treatments for non-small cell lung cancer, such as pemetrexed or the platinum doublet chemotherapy. But the disease does progress, and it occurs in the brain and is actually a key issue for crizotinib penetration. So to mimic this model, the non-small cell cell line, which is H2228, carries an EML4-ALK fusion and responds very nicely to ALK inhibitors like crizotinib, like alectinib, in these sort of subcutaneous or orthotopic implanted sites. But when you implant into the brain, you can actually see a vast difference in the response of the brain tumors in response to crizotinib and alectinib, and obviously you get really good tumor regression. So modeling this sort of tumor microenvironment is really important. As you can see that there's a lack of response to certain types of agents, which is a type of sort of resistance. So having understanding what resistant mechanism is needed is really important, again, when you are trying to select the right type of models. So in some cases, a subcutaneous model might be sufficient. In others, you may need an orthotopic model that is more representative of that tumor microenvironment. Okay, so in the next example, I'm going to show you the objective of that study was to identify drug resistance using patient-derived models in 3D and the impact of having stromal cells. So this diagram here shows sort of a typical cycle for PDX screening in vivo. So this would be obtaining the sample, creating the model, and then doing the screening with 2 different agents. You can also use PDX material in a 3D ex vivo culture, which is much faster and also more higher throughput for screening drugs or large collection of agents or combinations. So this approach was taken and was published as shown here on the right, and the difference in response in 30 non-small cell lung cancer PDX models was shown, and this is represented in this heat map on the right. So we reported that in some cases there was increased resistance that was observed when the cells were, the tumors were cold cultured with CAFs. And so here at the bottom are the differences in IC50s, responses to chemo in the presence of CAFs. And then the table shows the different combinations across the panels of non-small cell lung cancer PDX with and without CAFs. So there's a direct sort of comparison. And this model here, just to kind of highlight what that looked like in terms of IC50s, you can see here that, so this model is an EGFR has an EGFR activating mutation, has exquisite sensitivity to alatinib, but in co-culture with the CAFs, the IC50 is actually much higher. And then also in this study, it was reported that these models in combination with HDAC inhibitors, the resistance was reduced across those sort of models tested. So again, another different way of modeling resistance, which is based or driven by the cancer-associated fibroblasts, so an off-target resistance mechanism. So a lot of the models are already available and can be accessed, as mentioned. We have also have to create some models, and some of them already are being characterized, so they're not readily available at the minute. But these need to be created just because of the different types of resistant mechanisms that are emerging. So this next step is really important, because if you can't find the model within the collection already or in publications, the appropriate model needs to be created to mimic that sort of initial step 1 definition of resistance. And there are several strategies or solutions to achieve this, and these are broadly represented here. So first, you can look to acquire the models from the clinic, which we know is not always possible. So models can be created with either drug challenging in vitro or in vivo. There can be different ways of doing genetic engineering. Different sort of technical advances may enable some innovative models to be created. And this may include things like the different surgical models in the different tumor microenvironments or organoid technology. So these have been most successful strategies, and as you can see, these, as a result, we've been able to take these on board and expand our resistance capabilities quite significantly. And again, just listed here are more recent models that are in the pipeline being created or validated that reflect predominantly the agents, the new agents that have been approved. And where there is a need to tackle resistance. So, for example, the PARP inhibitors, RET has shown the ALK inhibition, KRAS, and then also PD-1 blockade by immune checkpoint inhibitors. So these anti-PD-1 resistant models are actually syngeneic models, which have been engineered with specific mechanisms to induce resistance. So just to kind of talk through about some of the rationale of that. So the mechanism of action of primary resistance to PD-1 blockade is largely unknown, but there are indications of STK11 or LKB1 alterations that have been reported to have a significant role in primary resistance to PD-1, PD-L1 axis inhibitors, and also specifically in the KRAS mutant lung adenocarcinoma. So this is sort of clinical information that is published. The loss of function of LKB1 induces production of different cytokines. This leads to neutrophil recruitment, which impedes T cell trafficking and activity. So it's important to identify treatments which can rescue the efficacy of PD-1 blockers in these STK11 mutant patients. And so what we did was we developed a STK11 knockout model using CRISPR-Cas9 technology. And the model used was a CT26 colorectal cancer syngeneic line, and this harbors a KRAS mutation. Because we don't have one with lung cancer, so we kind of opted for the CT26, and this has been used to evaluate the response to checkpoint inhibitor alone and in combination with MEK inhibitors. And so, as you can see here, the STK11 deletion was verified by Western blotting. Now, increase in tumor growth actually was seen. And also the loss of anti-PD-1 efficacy in the STK11 knockout models. Again, this is in direct comparison to the CT26, which is shown here. The combination of trametinib with anti-PD-1 produced significant anti-tumor activity, and then also a MEK inhibitor, another MEK inhibitor produced even better anti-tumor activity against the knockout model. And then flow cytometry, which is shown here, was used to actually profile the infiltrating immune cells. And this showed that with MEK inhibitors, there was a decrease in the GMDSCs, and which restored CD8 positive T cell infiltration, and also the anti-PD-1 efficacy in those knockout tumors. So this engineered model in an immune competent model was actually necessary for evaluating a specific mechanism of resistance in that fully functional immune system environment. So obviously this is a mouse system, so there are still some limitations, but it was adequate to answer specific questions and beneficial for sort of assessing the combination in that clinically driven mechanism that was engineered. So one important component of creating these models, as you've seen, is actually that step 4, looking to see what the characterizations of that model are to make sure that they do actually mimic the initial intention in step 1 as closely as possible, but also for the potential to look beyond just the efficacy testing and identify potential biomarkers. So there's many different analysis and services available that can be applied to in vitro and in vivo resistant models and profiling. And these lab services can be used to create baseline data for those models, which can then be correlated to the responder, non-responder profiles. And this then can be used for biomarker discovery. And actually, studies show that if those clinical trials which use biomarker strategy for patient selection are more successful when compared to those that don't. So really, you can apply the different types of analysis that are shown here, like the genomics, transcriptomics, proteomics, the immune profiling, for the relevant model types that have been generated so that you get good characterization of that baseline data. And then you can evaluate with bioinformatics support, especially when you've got large complex studies or large complex data sets, especially like proteomics or next generation sequencing, to determine those biomarker strategies for that particular agent or combination to overcome resistance or identify those patients who will benefit from that treatment. And just to give you an idea of the analysis undertaken for large screens, so this is an example of composite biomarker development for irinotecan response and resistance, which was just used as a proof of concept. So here the plots are showing the sort of distribution of a response of 256 cell lines using the area under a curve, which is according to sort of cancer type. And then reordered according to the sort of baseline in sensitivity or insensitivity, and then sort of the medium sensitivity. So you can see that this has been kind of split into sort of different sort of areas where you get responders and obviously non-responders being profiled. We did actually conduct a principal component analysis of the global gene expression data for all of those cell lines to kind of potentially identify a natural clustering of the groups, but really didn't find anything obvious between the sort of sensitive and non-sensitive models. And this is based on the sort of 20K human gene expression. The functional analysis by gene ontology, and this is of biological processes, was also done, and this identified DNA replication as having the most differential expression between responders and non-responders, and this is regardless of the number of top genes selected. And again, this is in line with the mechanism of action, so no real sort of novel finding. But what we did see was, and was quite interesting, was the SLFN11 was identified as a single gene that had significant expression for irinotecan sensitivity, and this is kind of shown in the middle. I don't know if you can see this, but it's shown in the middle. But it must be taken into consideration that having a single biomarker strategy is not always the best or achievable in many cases. And therefore may not reflect the sort of complexities of many of these diseases within cancer. So here the highest prediction or accuracy was modeled for irinotecan around a 21 gene set, which is shown in the sort of box here. And so here the data was then correlated with the area under the curve, and this so you can see that the signature score in all cell lines, including that sort of a middle group, correlated highly with the sort of composite biomarker in comparison to the single gene. So while the expression of the SLFN11 was the strongest single gene biomarker, the expression based on the 21 gene composite biomarker achieves a higher prediction accuracy, and so therefore there would be a recommendation. So for the final part of this webinar, I'll give an example of a model that was created to answer a specific mechanism of resistance following the 4-step strategy. And this has since been extensively used. So this is focused on the EGFR mutations and resistance in CDX models. And we actually have a number of different ones based on the sort of different mechanisms of resistance. And so the discovery of EGFR mutations has made a dramatic impact on the treatment options for patients with non-small cell lung cancer. And the EGFR targeted therapies have shown considerable promise, but drug resistance really is a substantial issue here. And the EGFR activating mutations, which have been targeted with first generation inhibitors such as gefitinib or erlotinib, And then there's been second generation irreversible inhibitors, but the emergence of resistance has resulted in further mutations, so like the T790M gatekeeper mutation, and that actually led to the fast development of osimertinib. Recently, obviously, there's been resistance that's emerged to osimertinib, and this has been reported to be mediated through further mutations, such as the C797S mutation. So there really is a need for relevant preclinical models to identify new strategies or combinations to overcome this third generation EGFR inhibitor resistance. So we have specific models that we've engineered for the triple sort of mutations in non-small cell lung cancer lines. But there are also other off-target mechanisms of resistance that have emerged in the clinic that need relevant models to represent this sort of patient population. And this, therefore, will then enable effective strategy to overcome those types of resistance as well. So the objective here was to see if we could model some of these sort of bypass signaling activation mechanisms as shown in this paper here. So in order to find the right model, the HCC827 non-small cell lung cancer is an adenocarcinoma line, and it harbors a activating EGFR mutation. So it's an exon 19 deletion and shows exquisite sensitivity to alatinib. And this was used to generate the EGFR inhibitor resistance via in vitro to try and see if we could mimic that sort of bypass mechanisms of resistance. Now, this case study is actually also available in our application note on this particular cell line, so you can also access this via our website. But I'll just show you some of the data here. So both alatinib and gefitinib were used to create the resistant variants by repeated exposure to escalating concentrations in vitro. So these two figures detail the response of the wild type line, which resulted in the generation of the alatinib resistant, which is denoted as ER1, and the gefitinib, which is GR1. And you can see that there is a 10,000-fold shift in the IC50 for alatinib and a 1,000-fold shift for gefitinib. And then the next step was to actually profile that resistance. So firstly, there was obviously no changes in comparison to the wild type model in terms of SDR profiling. So it was confirmed it was the same cell line. The lines were characterized for some of those mechanisms, so c-Met genomic amplification by PCR. We also looked at AXL gene expression and saw a significant increase in c-Met copy number and also AXL gene expression. We did also check the EGFR mutations. So we confirmed that there was still the exon 19 deletion in all the samples, but we didn't detect any further mutations in any of the exons, such as exon 20 T790M mutation. And then in vivo, the subcutaneous ER model, as shown here, was dosed with 25 mg per kg, so we've done always, which typically will show tumor regression in the wild type model. But here we actually see that there is a progressive disease rather than that sort of tumor regression, which is indicative of that treatment escape and resistance. To qualify if the resistance was driven by c-Met, combination study in vitro was conducted to determine the sort of synergistic potential of overcoming resistance and then restoring the sensitivity to alatinib. So different combination ratios were used in the parental line and also the resistant lines in a cell titer blue readout, and synergy was calculated using the calcusin analysis. And as summarized in the table, there was very strong synergy score observed with the combination, and this was then progressed further to in vivo evaluation. So here the mice bearing subcutaneous HCC827 or the resistant model were again dosed with 25 mg per kg, or a combination, with crizotinib alone at 50 mg per kg, or a combination of both, to demonstrate if this model could be used to show that sort of combination advantage. And so the graphs actually shown here are the percentage of the initial dose volume, just for comparisons between the two models. In the wild type line, there was no effect with crizotinib alone, which is shown by the yellow in comparison to the orange vehicle group. Obviously tumor regression with erlotinib, but in the ER1 line, crizotinib showed a partial response similar to the erlotinib treatment, and in the combination treatment restored tumor regression, indicating an additive effect and confirming the role of c-Met amplification in the resistant mechanism for this ER1 model. We also applied image-guided radiation using sort of protocols that you use similarly in the clinic. So this is showing the sort of combination strategy in those ER1 models to overcome EGFR inhibitor resistance. So this is fractionated radiation administered at 3 grays per day over 5 days. The wild type model was responsive to radiation therapy, but actually the ER1 showed some resistance to treatment, but this could be reversed with combination with crizotinib as well as erlotinib. This model has been widely used across different studies and combination approaches. So I'm just going to finish off giving some examples of some of the published studies. And in this one, which was in cancer research, the model, because it's a model of exon 19 deletion with MET amplification, was used to demonstrate durable response of a bispecific antibody which targets both EGFR and c-Met. Other models have actually also been used in this study, so it shows the broad response, including some of our non-small cell lung cancer PDX models. So the LU1235 harbors an exon 19 deletion, but shows partial response to erlotinib. And then you have the LU1868, which has a L858R mutation, as well as a T790M mutation, which is resistant to erlotinib. So again, just qualifying the response and the effect of that sort of bispecific. And then in a follow-up study published in Molecular Cancer Therapeutics, the resistance to third-generation EGFR irreversible inhibitor osimertinib was also observed as a potential. So there was a potential for resistance to this in this cell line as well, and also resistance that may be emerging for that novel dual targeting bispecific antibody was also evaluated. And this was done by mass spec-based quantitative proteomics and phosphoproteomics to understand the resistance that emerged by looking at the different signaling changes that occurred in those resistant tumors. And what they identified was that there was unique rewiring of tyrosine kinase phosphorylation, which was identified specifically for the EGFR family of SRC family kinases, so the SRKs, which allowed the tumors to escape that EGFR inhibition. Combination of osimertinib and SRK inhibitors was also shown in vitro and presented as a novel sort of strategy to overcome that potential resistance. And this was again nicely demonstrated in this study. This model has also been used to show potential strategies to overcome that osimertinib resistance in this MET amplified model, and this is in combination with a SHP2 inhibitor, and this was published recently in Cancer Research, and again it nicely shows here where the combination. So either it was administered early or it's administered later, showed a very good effective response and also restored the EGFR inhibitor induced regression that has been lost in these models. So hopefully I've been able to kind of show how valuable these resistant models can be. In this particular one, which is specific for the EGFR tyrosine kinase resistant variants, has been used across numerous different studies, showing different strategies that could be employed to overcome resistance through combination treatment. And then final slide. So back to this 4-step strategy and how this can be applied across multiple different types of resistance and models, which I demonstrated with some examples from our collection. There's not always easy to do. Sometimes it does take a bit of time to develop these models, but hopefully from our experience in resistant modeling, we can help tackle some of these high unmet clinical need and bring more benefit to the patients. So thank you for your attention and happy to take any questions.